All right. I wanted to continue talking about cholesterol a little bit. At the end of last time, we had gotten to, I think, this slide where we were talking about cholesterol. Cholesterol is a lipid. It is a steroid lipid. And that means that it has those 4 fused rings. So we know that it is a lipid if it's a steroid if it has that structure. Cholesterol does serve useful functions. You do need some cholesterol. You need some fat in your body. So some people will actually go on diets where they completely eliminate fat out of their diet, and that's not a good thing because you do need fat in your diet. You need it for hormones, and you need it also for cholesterol, as well as insulation and cushioning of your body. So you need fat. You just don't need lots and lots of it. Steroid cholesterol, what it will do is it will moderate the effect that temperature has on a cell membrane. So at warmer temperatures, it's going to keep the membrane from flying apart. The membrane is really only held together by the fact that there are water molecules that are keeping it in place. But if the temperature gets too high, the membrane can actually kind of fly apart. And that's shown here on the next page. The first picture is, let's see, I Googled how to make this pen size bigger. Let's see if it actually works. And it said just to do this. Let's see, is it bigger? Ha ha ha! Oh, I'm so excited. I'm way too excited. Yeah, okay, sweet. Nice, let's get rid of that then. So in this first one here, remember our membrane is fluid. It moves up and down and back and forth. And part of that is going to be because of these unsaturated lipids that we have sticking in the membrane. And those unsaturated fats are going to, because they kink a little bit, they keep the membrane a little more loosey-goosey, farther apart. The molecules are a little farther apart from each other. If you have a bunch of saturated fats, here I've got saturated fat, and there are none of those kinks. So it's very tight. It's very rigid. It's not going to move. It's not going to flex as well. So first having that unsaturated fat in there is important. And then also having these little cholesterol molecules. The cholesterol kind of does the same thing. It adds a little space or a little gap between the phospholipids. And that will keep the membrane from flying apart when it's hot. And it will keep it from getting too rigid and cracking and breaking when it gets too cold. So it serves both of those things. We do see that in species that are there's a big temperature change. You do see a difference in the cell membranes of those creatures, of those animals that are living in those different regions. And in general, you see if you're living in a place that's close to freezing, you're going to see that the cell membranes of those organisms are more flexible. They have more cholesterol in them. They have more unsaturated fats in them. Whereas if you look at organisms that live in a more stable environment with less fluctuation in temperature, either less heat or less cold, kind of moderate year round, then you see more rigid saturated fats because it's not a big deal to lose that flexibility. Okay, so we talked about last time we talked about how the cell membrane is a fluid mosaic. And fluid meaning that the phospholipids are moving back and forth. And then also mosaic meaning that there are all sorts of different proteins. They have different shapes, and they all do a slightly different function, and they're embedded in that cell membrane. So we call it a fluid mosaic model. Just like this little guy, this little bird is a mosaic made up of many different little tiles. The same thing with the cell membrane. It's made up of many different individual parts that are varied, they're different. And it's going to be different depending on what species you are. Like I said, if you're in hot or cold environments, it's also going to be different even within a certain species. You might see variability in terms of the membrane flexibility and what proteins are in those membranes, depending on what kind of cell it is, you're going to see different proteins. So there are proteins that are called peripheral. That means that they are bound to one side or the other of the membrane, so on the periphery. They're on the outside or they're inside. And those proteins, if they're bound to the surface of a membrane, are they going to be hydrophobic or hydrophilic? Have to be hydrophilic, right? Because they've got to interact with the water. So those are going to be proteins with a lot of amino acids that are going to be hydrophilic. You can contrast that with integral proteins. And integral proteins are ones that go through, well, I shouldn't say go through. They go into the hydrophobic region of the cell membrane. So again, if you are going to go through into that hydrophobic region, you have to have amino acids now that are also hydrophobic. Because if you have a bunch of amino acids that are hydrophilic, they will be repelled from each other, from the membrane. So you have to make sure that they match. There are also proteins that are called transmembrane proteins. Nope, wrong button. There we go. And these transmembrane proteins go through or span the entire length of the membrane. And in that case, you're going to have proteins that have both. They have to have some areas that are hydrophilic, interacting with the water, and you have to have some that are hydrophobic and interacting with the tails and the inside of the cell membrane. The hydrophobic region of the integral protein consists of one or more stretches of nonpolar amino acids. Those are often coiled into alpha helices. If you remember from the first unit, we talked about protein structure, and there was a secondary structure. And the nonpolar or the hydrophobic areas are often coiled like that as they go through the membrane. And that's shown on the next slide, where I have a transmembrane protein. And these bits here are going through the membrane, and they are all helices, showing that they're probably going to be the nonpolar, they're going to be the hydrophobic molecules, and they're going to be interacting with this inside, which is also hydrophobic. Is it? And then these guys out here and here, they're going to be the ones that are hydrophilic because they're interacting with the water either on the outside or on the inside of the cell membrane. I can't tell you how excited I am that my pointer is bigger now. It's the little things. There's lots of functions that proteins can carry out, and lots of functions that just the membrane in general carry out. Transportation, moving things across the membrane is important. Some of the proteins embedded in the cell membrane are going to be enzymes. There's also something called signal transduction, and there's a few others here. I'm just going to go through the list in pictures. And so transport, you can move molecules across the membrane. There are some that are enzymes that are embedded in that cell membrane, and their job is to catalyze reactions, but only if the molecule is at the level of the cell membrane. There's something that is called signal transduction. We're going to spend a lot of time on signal transduction. A little bit later, you're going to do a project on it. But signal transduction is basically somebody's knocking at your door. So out here, I have a signal molecule, and that signal molecule is hydrophilic. And hydrophilic molecules don't easily get across the cell membrane because once they get to the tails, they're repelled by the tails. And we'll expand on that a little bit later. But anyway, he's knocking and he still wants to get his message across. So this protein molecule here, its job is going to be to take that signal, that chemical molecule, and change it into a message that the inside of the cell can understand. And that's signal transduction. Sometimes there are proteins that their job is to recognize another cell. So I've got this protein here with the little rabbit ears. Again, purple is always protein. So the purple part is protein. The green part that you can't actually see because I erased it was this little green part here is a carbohydrate. That's why it's called, if you look at the text, it says a glycoprotein. And these are proteins that recognize, or I should say, are recognized by other cells. So if, say, an antibody is looking for a particular type of cell, it would be able to recognize that cell by the marker or the protein that is attached to the cell membrane. Say, oh, yep, this is the one I'm looking for. This is the spot. This is where I need to be. There are proteins that connect the two cells together. So they're just anchoring each other together. And then, like we talked about yesterday, there are also proteins whose job it is to anchor that cell to the surrounding environment so that the cell isn't moving around. And so my heart tissue all stays together and it doesn't start bouncing around, and you end up with your heart cells down where they're not supposed to be. Okay. All right. I think they're just showing these in a little more detail. Yep. So, cell surface proteins, these are the ones that are working as recognition molecules. These are important in medical. There's all sorts of medical applications. There's just one here shown as an example. HIV is the precursor. It's the virus that causes AIDS, and that compromises the immune system. What HIV does is it actually can recognize the markers on our immune system cells. And those are these surface proteins that are called CD4s and CCR5. You don't need to remember the details on this. But the point is that they're looking for those protein markers that are embedded in the cell membrane. HIV, they found that some individuals don't have that CCR5 receptor. And because of that, they don't usually get infected with the virus, HIV. And so they're resistant to it. So now there's people who are targeting making drugs that will allow the body to mask the fact that they have a CCR5 receptor. If you can trick the virus into thinking that it's not there, then it won't affect it. So it has to get into the cell to actually have an issue with the cell. So I think that's this piece. Yeah. So on the left-hand side, we have HIV. It's seeing those two receptors there, and once it binds to those two markers, it can be brought into the cell. So this guy's getting brought into the cell. It's like, sweet. I tricked him into bringing me in. I tricked the body's cells into bringing me in. This guy's like, nope, no, not going to work because you don't have this second receptor. That second receptor isn't there. So it's going to be, boop, sorry. Okay. So the more we learn about some of these illnesses, the more we can do to then, okay, well, let's see, can we do this or can we mask it? What can we do? So that's just an example of that. Cell-to-cell recognition. Cells recognize each other by binding to molecules, often to the carbohydrates on the extracellular surface of the cell plasma membrane. Membrane carbohydrates can be covalently bonded to either a lipid, and we call them glycolipids if that's the case. Or more commonly, they're actually bound to a protein, and we call those glycoproteins. Like I said a little bit earlier, the markers, collectively I call them markers, but glycoproteins, glycolipids, these molecules are specific to a species, which is one reason why COVID can infect some species, but it can't infect every species. In general, dogs, for example, and cats were they didn't have to worry about COVID because of the fact that they didn't have the right markers. They didn't have those right protein markers. Individuals, like I said, some people HIV affects and some people it doesn't because they don't have the same the same markers. And even when you look at individuals within their cell type, sometimes the cells just don't have that marker, and that's why when you get COVID, it doesn't. I shouldn't say this because there's so many strains now and everything's basically changing. But COVID tends to affect your respiratory system because those cells have the right cell recognition marker on them for the virus to be brought in. It doesn't tend to affect other organs. For example, it doesn't tend to affect the stomach, or it doesn't work on the pancreas or the liver. Now, I say that, but there are some strains now that are doing some funky things. I did have a friend whose dad got COVID, and he was having problems with his kidneys because now it's causing you to retain, or was it retain or get rid of? I can't remember which it is. Either retain or release too much fluid, and that can be fatal if it's not dealt with. So. Especially in older people, that the current strain of COVID is kind of an issue. Okay. Not quite sure why these slides are here. Let's see this. This is just kind of a review of when we're talking about proteins. And we're talking about membranes. Membranes have a distinct inside and an outside face. And because of that, the distribution of those mosaic pieces, those proteins, the lipids, carbohydrates, is going to be, you're not going to see them. You're only going to see them on one side of the membrane, not on both. And that asymmetry is built in when the membrane is actually built. Remember, the smooth ER is responsible for building lipid molecules, and then finishing them off in the Golgi. So when they leave the Golgi, they've already got that kind of orientation, so that they are already asymmetrical, and they can only be put into the membrane in one orientation. So they can only go on the outside, or they can only go on the inside of the cell membrane. All right. Questions so far? All right. When we talk about membranes, one of the things that is important is the idea that the membrane is selectively permeable. And you can think of selectively permeable as just a fancy way of saying it's picky about what it's going to let through. A cell has to get nutrients in, and it has to get waste out. But there's other things that we don't necessarily, maybe never want inside the cell, and sometimes maybe we only want it under certain situations. We want it inside the cell. So that's the selective permeability piece. A lot of that has to do with the fact that you have heads that are hydrophobic. Sorry, I said that wrong. You have tails that are hydrophobic and are hiding on the inside. And then you have hydrophilic molecules that are, that like the water, But the reason it sounds a little counterintuitive because it says that hydrophobic nonpolar molecules. Oh, that was not what I wanted. Sometimes that Control Z does not work. There we go. The hydrophobic or nonpolar molecules, so hydrocarbons, can get through the cell membrane. So hydrophobic can get through, which is a little weird. Whereas hydrophilic molecules, like ions and anything that's polar, it's hard for it to get across the membrane on its own. And before we go on, let me show you why that is. That's my head of my lipid, and that's my tail. Two tails. Here's another head, another tail. Yeah, I'll do one more. Okay. The reason that hydrophobic molecules can get across is because there's enough distance from here to here that they can pass between two heads and really not interact with those two heads. So even though they're hydrophobic and the heads are hydrophilic, they really don't come in contact. They can just kind of scoot their way through here. Then once they get to the tails, those are lipids. Those are nonpolar. They're hydrophobic. And they like the nonpolar hydrophobic molecules that are coming through. Obviously, they don't like them, but they're not repelled by them. They can interact with them. So my little molecule here that I'll make a little triangle, it can skirt through here and not be affected by the heads on either side. It's not going to be repelled by those. Okay. On the other hand, if I have I wanted black. If I have a molecule that is polar, he's like, hey guys, how's it going? It loves the heads, so that's not a problem. But then when it gets to the tails are like, we don't like your kind. We want you to leave. It's like sad face. So unhappy little polar molecule or ion. Ions, same thing. They tend to like the polar environment. They tend to interact well with them, and they're going to be repelled by it. So anything that's polar, long story short, anything that's polar needs some other way to get across the membrane. Anything that is nonpolar can get across the cell membrane. Okay. When I say polar, I'm talking hydrophilic. Polar is hydrophilic, and nonpolar is hydrophobic. Those are synonymous. Yes?
So what is that something else that it would need to get across?
So that's a brilliant question, and that's our next little bit here, which is there are proteins. So proteins act as a safe zone for those polar molecules to get across. And we're going to come back to this when we do cell signaling because that becomes a big issue. Some molecules need to get their message to the cells. Some of them can get across on their own without any help. Some of them are going to need some kind of messenger molecule to help them get across. And we're going to spend quite a bit of time talking about that. But that's for a later date. That's not yet. Right now, we just want to talk about transport proteins. So again, because these polar molecules cannot get through, they need a safe zone. And transport proteins have areas that are embedded in that hydrophobic zone that allow for anything that's hydrophilic to get through. So anything that likes water can get through because there's a little opening there. The protein has an opening that's made of many amino acids that are polar. And one example of that would be with water. You have a specific channel protein called an aquaporin. And aquaporins are just for movement of water because water cannot get across the cell membrane very easily. It needs help. So those channel proteins can open, they can close, they can let more water in, they can keep water from coming in, depending on the need of the cell. There are other transport proteins that are called carrier proteins that physically bind, attach to the molecule that needs to come in, and shuttle them across. And we'll talk more about that as we get into the course as well. Alrighty. Okay, so diffusion. Simplest way to get something through a cell membrane is through diffusion. It doesn't take any energy. And it's really just this idea that molecules, I like to use the fart analogy. Okay, if I fart, there's always that fear, right? It's like you want to run away from the fart because then no one will know, right? Except after a while it moves, right? Does it stay where it was? No, it moves. So if I let one rip, which one of y'all are gonna get to it first? People in front. Yeah, sorry guys. Yeah, I don't know. I released some wind here. Here we go. And at first, all those molecules are really. Can I do a butt cheek? Is that like butt cheek? There we go. That was pretty good. That's pretty good for me. Okay, so initially, all those smell molecules, all those molecules are going to be in the same location. I do this because you'll remember it, by the way. But over time, molecules are always in motion. There's always a little bit of motion associated with them, so they're going to spread out. So I'm sorry, you all, but you're going to get stuck with the smelly stuff too, okay, depending on how bad it is. Right? And then over time, it spreads out so far that you can't smell it anymore because it's below the threshold that's needed for you to smell that. Okay, but those molecules are always in motion. So molecules will want to move across the cell membrane if they can, and some molecules can use passive diffusion to move across a cell membrane. What did you learn in biology today? Well, we talked about farts. So here is, now we have a membrane. And we're just showing the molecules moving. And you can see that over time, they hit what we call equilibrium. So there's really equal numbers on both sides. And then if you have two solutes, again, that will kind of impact the space available. So the solutes will also move, and they'll try and get as far away from each other as they can. There we go. Substances will always move from where they are most highly concentrated. They tend to try to separate as far from each other as they can. And that is called the concentration gradient. So if I have my picture again, because I know you guys really liked it, there we go, releasing. That's my high concentration there. And they're going to want to move to a low concentration. So they're going to try and spread out. So when I say concentration gradient, when I say, and it's a misnomer, but it continues to be said. Substances will diffuse down their concentration gradient. That does not mean physically it will move from where it is high in the room to where it's low in the room. It only says it's moving from where you have a high concentration to a low concentration. So it could move from side to side. It doesn't have to move down, physically down. Okay, it's not like a gravity thing. When they say down, it's concentration gradient. The biggest, I guess, thing that you need to know about this would be the fact that this is passive transport. It does not require any energy to do this. These molecules are already in motion. They're already going to try and spread out as much as they can. So if a cell can do this, this is the way that it really wants to do it, because that means that they're out nothing. They can easily get something across, and there's no effort, there's no loss to that cell. Unfortunately, that's not always going to happen. We're going to talk more about that. Not today. We'll talk about that next time. And I think I'll just talk about this for a second here. Osmosis is the movement of just water across a picky or a selective permeable membrane. It's diffusion of water, but you also add in that membrane in there. Water will also want to move in a certain way. Now, oftentimes it is said that water will move from a region of low solute to a region of high solute. So low solute to high solute. Well, we haven't talked about that yet. Solute is the dissolved substance that you find in water. And it could be lots of different things. It could be salt, could be sugar. And it will try to even itself out, because typically the thing that's dissolved in the water is too big to move. So what will move instead is the water. So the water will move and not the solute. If you have a little bit of solute, what does that mean about your water concentration in regards to the other side? If you have low solute, it means that side also has high water. Okay, high water. So let's say that high H2O. And my higher solute, what will it have? Lower. It'll have lower. I should probably say lower and higher. So where will my water move? To the higher solute, which is also the lower water. So it's just saying it, it's just one way of saying it versus another. Okay. All right. I am going to stop there.